welcome back to Biotech Academy's Immunology 101 lessons, and in today's video we will talk about the adaptive immune system. Let's get started. Alright, in today's lesson we will talk about the adaptive immunity and how it differs from the innate immunity. So basically we'll answer the question of what is the adaptive immunity. Then we will talk about the major cell types, for example T and B lymphocytes. We will talk about receptor systems that give adaptive immunity its specificity. And lastly, we will talk about how innate immunity and adaptive immunity are integrated into one seamless defense network. Okay, let's start with the first topic then. Unlike the innate immune system, which responds quickly but in a more generic way, the adaptive immune system is characterized by specificity, diversity, memory, and self-non-self discrimination. So, it is specific, diverse, can create memory for the future, and distinguishes between self and non-self. So why do we need the adaptive immune system? Well, the innate immune system provides the first line of defense. So it relies on pattern recognition receptors that recognize pathogen-associated molecular patterns. However, microbes evolve rapidly. They can alter their surface proteins and even inhibit phagocytosis. The innate system cannot adapt this fast. That's when the adaptive immune system comes into place. It has the capacity to generate an enormous repertoire of antigen receptors, each with unique potential epitope matches. But don't worry, we will get into detail, okay? This receptor diversity is caused somatically, so there is gene arrangements, for example, VDJ recombinations, which we will also talk about very soon. So here, the main thing you have to know is diversity in antigen receptors. Right. So in order for us to be able to understand the adaptive immune system, we have to talk about the key cells of the adaptive immune system. Firstly, I will talk about B lymphocytes, and then I will explain you T lymphocytes. So let's start with B lymphocytes. B lymphocytes, each B lymphocyte expresses a B cell receptor. A B cell receptor. So this is our B cell, and on top of it there is a Y-shaped receptor, which is a membrane-bound form of an immunoglobulin, aka an antibody. So if we ask you what a B cell receptor is, you say it's membrane-bound, and you say it's an immunoglobulin, aka a type of antibody. And now you know what a B cell receptor is. I would like to talk about the structure of the B cell receptor. This section here recognizes the antigen. So antigen is the part of a microbe. This one is the antigen. And the B cell receptor can bind to it, right? To recognize it. And this exact part is binding on it. That's why it's called the recognition site for an antigen. We have heavy chains and light chains. These chains in the middle that I am showing you right now, these ones, are heavy chains. And the ones outside are light. Then this part is the part that is bound to the membrane. The variable regions at the tips of these chains form the antigen binding site. Important, antigen binding site. Specifically this region here, this region is CDR. Complementary determining regions. When a B cell receptor binds to a specific antigen and receives help from a T follicular helper cell, it becomes activated. It then differentiates into plasma cells. So the B cell turns into a plasma cell, which secretes soluble antibodies and memory B cells. So antibodies are the immunoglobulins that are roaming around freely in our body. And this causes long-term or a faster response to future infections. Let's talk about T lymphocytes. These are central players of the cell-mediated immunity. So each T cell expresses a T cell receptor similarly to the B cell receptors. The receptor is a heterodimer made of an alpha and a beta chain. And in some cases, it would be a gamma delta chain, which is a smaller subset. So unlike B cell receptors, the T cell receptors cannot recognize free antigens. So no free antigens. They only recognize peptide fragments presented on MHC molecules. This is going to be important. So MHC molecules are found in our immune cells, and those other immune cells are basically presenting the antigens to the T cell receptors. Okay? So, as you remember, we have MHC class 1 molecules, which present peptides from intracellular proteins to CD8 plus cytotoxic cells. And then there are MHC2 molecules, class 2 molecules, which present peptides from extracellular proteins to CD4 T helper cells. So we will get into detail about the CD8 and CD4 cells, and later I'll also explain you the MHC1 and MHC2 molecules. So let's talk about the subsets of T cells. There are CD8 positive cytotoxic T lymphocytes. CD8 is a type of receptor, and if the T cells have that receptor, we call them CD8 positive cells, T cells. The same goes with CD4 positive T cells, which we call T helper cells. That means that the T cell possesses a CD4 receptor. If the T cell possesses a CD8 receptor, we call it a cytotoxic T lymphocyte. So a CTL in short. These can kill virus infector or transformed cells by releasing perforins and granzymes to trigger apoptosis. Now, on the other hand, if a T cell possesses a CD4 receptor, we call them T helper cells. And these helper cells are responsible to release cytokines. Cytokines are chemicals which are there to help the immune system see that there is a potential threat. So there are some subsets of T helper cells. For example, there are T helper cells 2, T helper cells 17, TFH cells, TREGs. For example, T helper cells 1 can drive macrophage activation, so to say intracellular bacteria and viruses. T helper cells 2 can promote B cell help for antibody production. So you can associate this with IgE production and B cell activation. Lastly, there's TFH cells. They reside in germinal centers and are essential for B cells, class switching, and affinity maturation. And there's something called TREGs. TREGs maintain tolerance and prevent autoimmunity by secreting interleukin 10. I know it sounds crazy right now, but we will get into detail later. So most importantly in this chapter is that you know that cytotoxic T cells and T helper cells exist as subtypes of T lymphocytes, and know that cytotoxic T lymphocytes have a CD8 receptor, whereas T helper cells have a CD4 receptor. Let's talk about specificity and diversity in our adaptive immune system. One of the defining features of the adaptive immune system is its specificity. Each lymphocyte recognizes a single molecular shape or antigenic epitope. You have to understand that, so to say, enemies in our body have to be recognized somehow. And we do that in the shape of epitope recognition. So epitopes are basically small protein sections of an enemy in our body. So when it comes to the potential of diversity, in humans we're talking about 10 over 11 unique receptor specificities. And this comes from combination diversity. So we have a long gene section, right? And we are able to take specific genes, specific sections, and we combine them. Mostly this is V, D, and J gene segments. And as said, there are multiple of them. And imagine I take one gene here, one gene here. This is the first combination. If I take the same gene here, but a different one here, that's another combination. And then I take the same one here and take this one, that's another combination. But let's say I don't want the first one, I take this one and this one, that's another combination. So all of these combinations result in this number. So when we talk about combination, you can say combinatorial diversity. Then there's junctional diversity. So random addition or deletion of nucleotides at the joining sites. So basically, at these sites where we bind it together are nucleotide deletions. Then there is combinatorial pairing, random pairing of heavy and light chains. So imagine you have created a light chain, right? There are different light chains, and then there are different heavy chains, and you just combine a random one with it, which causes also some type of diversity. I'd like to talk about somatic hypermutations too. So after activation, point mutations in the variable region's genes further refine binding affinity. A process called affinity maturation mediated by activation-induced deaminase. This means that in these binding regions, which are super specific, there are also point mutations happening, which further induces the specificity. This is called affinity maturation, and the enzyme responsible for this, the enzyme which causes these mutations, is called activation-induced deaminase. So if your teacher ever asks what causes specificity and diversity in these receptors, you can list these 4 main reasons. Let's talk about teamwork. It's critical to understand that the adaptive immunity doesn't act on its own in isolation. It depends on the innate immune system for activation and guidance. So if the innate immune system cannot completely fight off a bacteria, a virus, a pathogen, so to say, it activates the adaptive immune system. And the biggest player here are dendritic cells, a bridge between the adaptive and the innate immune system, okay? Dendritic cells are the professional antigen-presenting cells. So a dendritic cell is a type of antigen-presenting cell. They detect pathogens using TLRs and other PRRs. So TLRs are toll-like receptors and PRRs are pathogen recognizing receptors. So the dendritic cell first detects a pathogen via its TLR and PRR receptors, and then it presents that pathogen in form of an antigen through its MHC2 receptors. So when the DC cell gets a signal, aka it finds a pathogen, it gets activated. Dendritic cells migrate from tissues to secondary lymph organs like the lymph nodes or spleen, okay? And here in the secondary lymph organs, they represent peptides via the MHC molecules. In our case, an MHC2 molecule. There are some activation requirements. So in order to activate a T cell, the T cell receptor has to be able to bind to the antigen represented by the MHC receptor. Then there's a second signal. The T cell has a little subreceptor called CD28. That has to bind to the B7 receptor on the dendritic cell, aka it's also called a CD86 receptor. Cytokines play an important role too. These direct the differentiation of the T cell into Th1, Th2, Th17, and so on. And without these innate derived signals, the T cells becomes anergic. So it's a mechanism that enforces tolerance and prevents accidental activation by harmless agents, meaning you don't unnecessarily have an immune reaction. So here are some examples I'd like to give you. So you know that toll-like receptors were found on the surface of antigen-presenting cells. Toll-like 4 receptor can recognize bacterial lipopolysaccharides, which then lead to the production of interleukin 12, which then causes Th1 cells to activate and differentiate, like T cells to differentiate into Th1 cells. Then there is the helminth recognition, which triggers the interleukin 4 production, which then favors Th2 responses. Then there's fungal detection via dectin-1, and this promotes, for example, interleukin 6 production and TGF-beta, which again causes Th17 responses. Lastly, I want to talk about some clinical examples. In this case, I'd like to talk about the influenza infection. So the innate immune system first senses viral RNA via RIG1, and then this is triggering the interferon release and dendritic activation. The dendritic cells then migrate to lymph nodes. The dendritic cells migrate to the lymph nodes, and they present the influenza peptides via MHC1 and 2. So they present basically parts of the virus to other cells. Then CD8 positive cells become cytotoxic T lymphocytes that kill infected epithelial cells in the lungs. And the CD4 positive cells and T helper cells, so to say, help B cells produce high affinity neutralizing antibodies. So these go to B cells, and the B cells produce specific antibodies, which prevents infection. And after the clearance, memory T and B cells remain, forming the basis of long-term immunity and vaccine protection. So, in summary, the adaptive immunity is specific, diverse, and capable of memory. So, B cells mediate humoral immunity via antibodies, and T cells mediate cellular immunity via cytokines or cytotoxicity. Adaptive receptors, for example, B cell receptors and T cell receptors, arise from somatic gene rearrangement. So, gene rearrangement. The innate immune system provides the context and activation signals that shape the adaptive outcomes. So, the innate and adaptive immune system are tied together, they're connected. Lastly, the cooperation for both systems ensure effective, regulated, and lasting protection. So this was it for the video. Thanks for watching. Please like and subscribe and share because I'm putting really a lot of effort into this. It would mean the world to me. We'll see each other in our next video. Bye!